Hello and welcome to another video. We are back with the normocytic hemolytic anemias. This time we're going to be talking about the extravascular ones. So that's PKD, hereditary spherocytosis, and sickle cell disease. Again, from last video we spoke about these two overarching points that cover all of the hemolytic normocytic anemias that we're going to be talking about. So let's get right back into it. So with hereditary spherocytosis, we're looking at a mutation of the cytoskeleton of the red blood cells. So that's stuff like ankyrin, spectrin, etc. These are the specific mutations that occur. But we're looking at damage basically of the cytoskeleton. So the cytoskeleton is what is keeping the shape of the red blood cell. So if you think of like the human skeleton, how that gives us a shape and that holds the shape together of our body, it's the same thing with these red blood cells. So they have a cytoskeleton that helps keep their shape. But if we have a mutation here where that's not functioning anymore, and you can see in this image right here how it's not functioning anymore, then the shape of the red blood cells changes and they lose elasticity. And here you can see the spherocyte, what that looks like, and it's called a spherocyte because it literally looks like a sphere. So what happens is now that you have the defective red blood cell that is abnormally shaped, it gets trapped by the spleen because it's, remember, reduced its elasticity. And you see in this image right here how that happens. So the normal red blood cells pass through the splenic sinusoids just fine, and they get sent into circulation because they're healthy. However, these spherocytes get eaten up by splenic macrophages, and they don't fit through these splenic sinusoids, so they get destroyed, and that's where the hemolysis occurs. And that leads to, again, splenomegaly, because the spleen is being overrun, overworked, lots of red blood cells being destroyed, and so, of course, you get increase in unconjugated bilirubin and jaundice because you have all of this hemoglobin being released, right? That's in the red blood cell, and all the destruction of the red blood cell. It releases a lot of unconjugated bilirubin, and that leads to the jaundice. And so lots of red blood cell destruction, like we said before, because the spherocytes don't fit through the splenic sinusoids. Treatment would be a splenectomy because there's not anything necessarily wrong with the spherocytes themselves, but it's the process that they undergo in the spleen that's giving us our anemia. So if we remove the spleen, the patient should be okay. But now we have the emergence of Howell-Jolly bodies, which will always happen post-splenectomy. And the reason is because these are leftover DNA fragments that you see here in this image. And normally, the spleen will remove these in normal red blood cell production. But now that we don't have a spleen, they reemerge. So PKD is a Pyruvate Kinase Deficiency. This is a mutation in your PKLR gene. And what happens is that it results in a defect in the Pyruvate Kinase enzyme that halts glycolysis. So if we remember way back when in biology, glycolysis is one of the fundamental steps in creating cellular energy, also known as ATP. This process is going to form pyruvate, and then that's going to go on to different cycles to create ATP in the cell. So because we're halting glycolysis, now the red blood cells have reduced ATP production. And if you remember the sodium-potassium pump, that requires ATP, and so that stops working. And now we have an electrolyte imbalance in the cell, which is a major issue, and that's what causes the red blood cell death. So the pyruvate kinase enzyme is indirectly causing the hemolysis because it has a downstream effect of halting glycolysis, reducing ATP production, halting those sodium-potassium pumps, and causing a electrolyte imbalance. Big red flag for this would be a patient having a history of chronic hemolytic anemia, and PKD is actually going to cause these cells to appear. Those are called echinocytes, also known as Burr cells. And you also see reticulocytosis, like we see in all of these hemolytic anemias, where we have an increase in reticulocytes as the bone marrow tries to replenish the red blood cells being destroyed and lost. Again, we have splenomegaly because of the red blood cell destruction. They get stuck, like we said before, in the spleen, and they lyse, get destroyed, release a lot of unconjugated bilirubin, and that leads to jaundice. Treatment here will be transfusion and a splenectomy. And like I mentioned before, anytime we have a splenectomy, we should expect Hawa-Jolly bodies to emerge, because normally the spleen removes them, but without a spleen, we're going to see them come back. In the previous video, we spoke about G6PD and we spoke about PNH, so go ahead and watch those videos if you want to learn about those. And then lastly, we're going to talk about sickle cell disease and sickle cell trait. So, first, sickle cell disease. Sickle cell disease is a point mutation that happens at the beta globin gene on codon 6, and it's a single amino acid switch up from glutamic acid to valine. And what happens is that your normal hemoglobin A, which if you remember was 2 alpha 2 beta, gets converted to hemoglobin S. So now you have these sickled shaped cells that appear. Instead of having your normal red blood cell, they turn into sickle cells. And what's interesting about sickle cell disease is that it's actually asymptomatic until there's a physiological stress that triggers it. And it's stuff like high altitude, hypoxia, acidosis, infection, stuff like that. So those are the things that are going to trigger hemoglobin S to polymerize and cause these sickled shapes. So hemoglobin S doesn't have the structure to maintain its shape during these stressors. So what happens? It polymerizes, it plugs up small vessels. This recruits neutrophils that get activated. That, of course, causes an inflammatory response. And then you have something called vaso-occlusive crisis, which is very dangerous in sickle cell disease, as well as these things right here. So chest pain can occur due to the sickling of the red blood cells. You can have hemolytic crisis and splenic sequestration. So again, splenomegaly can occur and all those kind of things with the spleen, because these cells don't fit through the splenic sinusoids. That causes a lot of damage, causes the spleen to be overworked, destroys the cells. So we get this hemolytic crisis. And then we get aplastic crisis if the infection is parvovirus B19. Now, why is that? So normally in a healthy person, if they were to be infected with parvovirus B19, you wouldn't necessarily notice the dip in red blood cells, because the progenitor cells are being destroyed minimally. And red blood cells normally live to 120 days. This kind of destruction of the progenitor cells really going to last about a week on average. So you won't really notice it, because you have a lot of red blood cells, you're constantly producing them, and they're living to 120 days. So even if you're killing off some of the progenitor cells, some of the reticulocytes, it wouldn't be that big of a deal, because your red blood cells are living to 120 days. So if that's happening for about a week, you can recover from that. You may have a little bit of a dip, but it won't be life-threatening, as long as you, of course, treat the infection. However, if you're an unhealthy patient with sickle cell disease and you get parvovirus B19, this becomes a major problem, because your red blood cells are not living to 120 days. They're really only living 10 to 20 days. So if you get infected with this virus, your marrow stops producing red blood cells, because it's attacking the progenitor cells and all that kind of stuff. There's no reticulocytes. You have no reticulocytes to replace your dead red blood cells when the 10 to 20 day mark comes. And a lot of times, on average, could be a week, could be a little longer, that this virus is attacking those reticulocytes, and that becomes a major problem because now you can't replenish the red blood cells you lost. And so you get the aplastic crisis, which is basically your bone marrow stops producing red blood cells, and your red blood cells are already living such a short lifespan. It is a major problem. And I did forget to mention with the chest pain over here, this is speaking to something called acute chest syndrome, which is something you can see on x-ray, something you can see in sickle cell disease patients that has to do also with the vaso-occlusive crisis. This can occur anywhere in the body. So pain anywhere in the body should be a major red flag with sickle cell disease, especially after maybe a hike at a high altitude or having an infection, stuff like that. And then lastly, sickle cell trait is basically someone who has sickle cell, but they just don't have the disease. They're just carrying it. So this person, instead of the hemoglobin A being converted to hemoglobin S, they are kind of almost, I guess, like heterozygotes where they have both hemoglobin A and hemoglobin S present. So as long as they have less than 50% of hemoglobin S, so that is to say, as long as their hemoglobin A predominates their hemoglobin S, they're less likely to have symptoms. And these people especially have resistance to Plasmodium falciparum malaria, which is how people think or suspect that this came about, is that carrying that trait brings on this added benefit of malaria resistance. And that's why it's actually more commonly seen in people of African descent.